Alright, this is Mr. McMurray, and we're talking about the diffusion lab, which you hopefully have watched the video that we did yesterday showing kind of the results. Now, we did not talk about this one right here with the mass of the lab. Let's just say, I know that, I'm not sure if this is the one that we looked at, but one person had 10.7 grams, and then after it was weighed, it was 11.0 grams. And that was Caitlin and Alexis, and I think they did a good job, so that's probably good results to use, all right? And so when they first put their cell into the distilled water, it was 10.7, but after we took it out the next day and been sitting in there, it actually gained weight, okay? Now, one asks, how did the mass of the cell change, okay? And yes, it did. And then if so, how, we would say it increased or decreased. In this case, it increased. Now, why could it have increased? Well, we'll talk about that. So let's come back and look at our test, okay? If we tested for Benedict's solution, that's when we put the blue solution in and heat it up. Was it positive or negative? So that's the first thing, you would put positive or negative in the blank. And then after that, you would tell what color it turned. Okay, you may have to go back and look at the video again. That's okay. All right, so what color it turned? And I believe, if I remember correctly, we heated it. It turned yellow, and that was a positive test, okay? The iodine test for starch was a negative. It turned kind of honey-colored. Did not turn the dark black, purple or black that it would if there had been starch present. And last but not least, silver nitrate solution, or the salt test, to test for salt, was where it turned white, and yes, indeed, it did turn white, and it was then a positive test for salt. So what does that tell us? Well, it tells us that sugar had escaped from the cell, so it had either leaked out or it went through the cell membrane. Since everybody's showed up on the outside and not everybody's leaked, we assume that it went to the outside because it could go through the dialysis tubing, which was our cell membrane. And the same thing with the salt. Now, the starch did not. Okay, it still stayed inside the cell. So let's go back here. If our gained weight, well, it couldn't have been because the sugar left or the salt left. That'd make it weigh less. So something must have also come in, and since it couldn't have been starch, it stayed in, and obviously these things were leaving, then it must have been water. Exactly. So water must have been coming in and diffusing into the cell, and that's why it weighs more. All right. Now it says, so which substance tested positive? So that would be, you can answer that. Which one was negative? We talked about that. All right. Is the dialysis tubing selectively permeable? And the correct answer is yes. It did let some things through, but not everything, and that is the definition of selectively permeable. Now, it's not nearly as selective about it. It mostly has to do with the size of the holes, and therefore, why would we think starch did not? If you remember, starch is a whole bunch of glucoses hooked together, a big old long chain, and so it probably is not going to fit through the little holes in the dialysis tubing, whereas the small sugar and the salt molecules would. And so in this case, it was selectively permeable, but what may have affected it? Primarily probably the size of the substances. Now, it could also have been affected by temperature. If we had different temperatures, some was warmer than the other. There's a lot of other things that could have affected it, but probably the biggest one is the size of the holes. Now, the question is this active or passive transport? Well, active would require energy, and did the dialysis tubing have anything that would supply energy? No, there are no mitochondria in our made-up cells, so this would have to be passive transport, just going through the pores. All right. Now, so why was it necessary to rinse off the outside of the cell with distilled water before we put it in the cup? Well, if we'd gotten stuff on the outside of the cell, some of the sugar water or the salt water or the starch, then when we put it in the distilled water, that stuff on the outside would have just come out into the water, and that would have contaminated the distilled water. So we wanted to rinse it off to make sure that the only way that stuff could get out into the distilled water is if it went through the membrane, not just stuck on the outside of the cell. All right. Does osmosis occur? Well, we know it probably would because we had it in pure water, so probably water is going to be going from high to low, so it's probably going to move in. And in this particular case, it actually gained weight, and that's about the only thing that could have moved into the cells. So yes, osmosis did occur because water must have moved into the cell to make it weigh more. Now it says, did diffusion occur? That just means things going from high to low. Well, obviously the water, which is a type of diffusion, but we know that the sugar moved out, and we know that the salt moved out, and therefore they must have diffused through the membrane. All right, and so basically you just need to draw a picture here of your cell and show the stuff inside of it, which was the starch, the sugar, and the salt, and show which ones went out, which ones didn't. Also show what moved into the cell, if anything moved into the cell. All right, definitions. Obviously, you need to stop this if I'm going too fast. That's okay. Just stop it, and yeah. Some people wish they could do that in real life, but all right. Hypotonic solution is solutions that have less than. Hypo means less, so they have less solutes dissolved in them than the actual cell did. So hypo has less stuff dissolved in it. Isotonic, iso means equal, like isosceles triangles have two equal sides, and so an isotonic solution has equal numbers of solutes in the solution and in the cell. They're the same. Now, that does not mean that nothing goes in or out. That means about equal numbers of salt or water or whatever will go back and forth between it. Okay? Hypertonic, hyper means more than, so in this case, this solution has more solutes dissolved in it than the cell would. Okay? And therefore, water is higher concentration inside the cell and will go out, and salt and things like that on the outside will try to move in. All right, and the last one is intravenous or IV solution. Intra means into or inside a vein. So this is a tube that's put inside a vein with a solution that goes directly into the bloodstream. Anytime you go to the hospital for surgery or any kind of procedure, they will usually put an IV tube in. Why? Well, several reasons. One, if you need more water, you don't get dehydrated, things like that. They can also put nutrients in there. People who are in comas and stuff like that that can't eat and so forth, then they can put nutrients in their blood directly through an IV solution. Also medications, if they want you to go to sleep, they can anesthetize you. Yeah, the anesthesia will go through the IV. Medicines, stuff like that, they can put it directly into your bloodstream, so it speeds it up and makes it happen a lot faster. So that's what an IV solution is. Now, a couple questions about IVs. If you were in the hospital, say you're in the hospital, your feet were swelling, you have a lot of swelling in your body, you're retaining water, what kind of solution would be best? Well, it's got to be pretty close to isotonic, but it would probably be slightly hypertonic. That means it has more stuff dissolved in it, and that would encourage stuff to come out of the tissues into the IV. Okay, so that might encourage some of the swelling to go down. If someone is dehydrated, on the other hand, and there's not enough water in their cells, then you might use a hypotonic. Hypotonic means there's more water in it and less solutes, and so that might encourage water to go into the cells, which would help the dehydration. All right. Now, number 12 says, what about things like Gatorade? Would they be hypo, hyper, or isotonic? Well, it turns out they're actually about isotonic. They're about the same as your body would be, because the idea is if you need Gatorade, that your electrolytes and the things dissolved in the water are lower than normal. So if they put an isotonic solution, then some of those electrolytes will diffuse into your cell. Whereas if you drink just pure water, it would not encourage that. So they're usually actually isotonic. All right. Most IVs in the hospital are just going to be isotonic because you don't really want to change the concentration most of the time. We talked about if you're dehydrated or swelling, then you might do something differently. But in general, you're just going to keep it kind of isotonic. All right. Interstitial fluid, the fluid around your cells, are going to be roughly isotonic, about the same. There's going to be some things that are a little higher or lower, oxygen, waste products and stuff like that coming in or out because of the difference in that. But they could be either slightly hyper for some substances and then be slightly hypo for other substances. All right, concentrated salt water would be highly hypertonic. All right, now just for the record, C, the ocean, is supposed to be fairly similar to our concentration of salt and water in our body. It's kind of interesting. But if you had really concentrated salt water, like you might gargle with or something, that's definitely going to be hypertonic. Distilled water means there's nothing dissolved in it, so obviously that's about as hypotonic as you can get. Just for the record, water rarely stays hypotonic for very long because even gases in the air, carbon dioxide and stuff, start to dissolve in it and make substances. Alright, the attempt by cells to keep the same concentration of water and solutes inside and outside the cell is an example of body attempting to maintain, that's right, homeostasis. Keeping things the same is a big, big deal. Alright, let's go to the other drawing just so I can draw on it a little bit. But if you look at this, alright, here we have shows more water, the clear ones out here, than there are on the inside, so water is going to move in. So that means if there's more water on the outside, then that means it is hypotonic. Alright, so for hypotonic we would have A, and if all this water is going in, it will make the cell swell up, so it's going to look like that one, so it's going to be A3 or 3A. Alright, for this one, where it's about the same on both sides, technically there'd be stuff moving in and water moving out, but it'd be about the same. So it's just going to look normal, which would be this one. So that's going to be one. So sometimes you just have to be one. All right. And finally, the last one. This has a lot more solutes afterward. It's hypertonic, and the water is going to leave or shrivel it up. So you're going to have C, and that's going to look like number 2. Okay. So you can check your answers. Make sure you understand those because they will be on the test. So a test will be either Thursday or Friday, probably Friday to be honest with you. Alright? Alrighty, well, we will see you later. I hope you enjoyed this, and next thing up will be the test.